So good evening, everyone, and welcome to the second of 4 lectures for the fall of 2017 put on by SoMAS, School of Marine and Atmospheric Sciences, and specifically here in Southampton. I'm Chris Gobler. I'm the Director of Academic Programs for SoMAS here in Southampton. And I'm very excited tonight to be introducing Brad Peterson as our speaker. But before I give my introduction, I'll point out that we have two more editions of these seminars this fall. On Friday, November 3rd, Professor Joe Warren will be here, and he'll be talking about, I guess he's talking about marine mammals. Who knew? Yeah. And then another great from Southampton, Chris Paparo, will be giving our final lecture. That's going to be on Friday, December 8th. Which is the second Friday in December, and that's actually going to coincide with both the windmill lighting here, and I believe actually SoMAS is going to have a satellite 50th anniversary celebration on this campus on that same day. So save the date, Friday, December 8th. So with that in the books, I can introduce Dr. Brad Peterson. I've known Brad for a long time now. I was actually on my way in, just recounting. So 15 years. I hired Brad Peterson here. Maybe my greatest academic feat of all time was to bring Brad Peterson here as a marine ecologist in 2003. I had to convince him to come, and he did. And we've had a lot of really fun interactions through the years on many different levels, educational and research. And it's been great. And I really admire Brad Peterson for many reasons. He does great research. He has a great team of researchers. I really admire, he's a great mentor to all of his students. It's very clear that he's, in addition to being a great mentor, there's great camaraderie amongst his lab. I love, for example, this summer, when his lab had the end of the summer celebration, they surprised him with a t-shirt. It's Breaking Brad. and for those of you who know the show Breaking Bad, it had the silhouette of Walter White, bald head with a Fedora hat, and kind of bared a nice resemblance to Brad. And then I also remember we went through a very interesting time, 2004, when LIU was closing, and we had a pretty uncertain time. And it was almost time. I'm going on too long, but this is okay. It's a short crowd. You're going to like this story. I think I have my age about right. So in May, somewhere around May of 2004, we both had something very exciting happen in that I got tenure here at LIU, static. And Brad got an offer to be a professor, I think it was at Auburn University, same exact time. And so, at the time I'm running the marine science program, and so I get tenure, I'm very excited about that. And I hear Brad might be leaving. I say, oh, Brad, things are going really good here, and I said, and I remember the line. He's gonna laugh when I say that. I said, Brad, the best times are just around the corner. The best is yet to come. And so Brad begrudgingly says, all right, I guess I'll stay. Well, about a month later, we get an email. I remember the date, June 15th, 2004. We regret to inform you we will no longer be having a college campus in Southampton. And I remember the panicked phone call I got from Brad Peterson and Joe Warren. Gobler, you better come over here right now. Can you re-explain the best times are just around the corner? But true story, okay? And then we had about, I don't know what it was, but the next several months were exciting. It was an exciting time. And then transition started very exciting, and we did it together. And in hindsight, it was fun. But here's the thing that I thought about. At that time, when I said the best times around the corner, he joked about that. And I said the best times are ahead. And and we kept it as a going joke because we wanted to pull the ripcord and get the heck out of here. And he had the opportunity to do that, and he didn't do it, partly because of me. But in hindsight now, 10 years later, I think he would agree that actually, of course, I have no idea. But I think, things have worked out really well for Brad Peterson, and he's a lot to be grateful for, and he's had made incredible accomplishments. And so I'm excited to hear about all of them tonight. Too long of an introduction, but thank you for indulging me. So with no further ado, Dr. Brad Peterson.
While Chris is going back to his seat, I'm going to take a moment to remind him to turn his sound down on his phone. I know the Yankees are about ready to start.
Oh, yeah, that's right. Better hurry.
So, just like you have your own family tree, us as academics, we have our own family tree. And I want to share this with you because in the last 5 years, whenever I have an opportunity to give a talk to the public, I end up giving a talk about seagrass. And so people think I'm a botanist, okay? But my advisor was a guy named Ken Heck. And Ken Heck spent his entire career looking at how habitat complexity, three-dimensional structure, affected predator-prey interactions. And his advisor was a guy named Daniel Simberloff. And Daniel Simberloff was looking at how communities came together, how species were like puzzle pieces. And when one puzzle piece came in, one species, it would either make it easier for another or prevent a species from coming in. And of course, Daniel Simberloff's advisor was E.O. Wilson. And he was all about the ants. Remember? The ant guy. And so all three of these guys, just like me, were very interested in how species interact with one another. Not plants. But whenever I get a chance to talk to people, I talk to them about this amazing habitat that we have, seagrass. Now, my guys in my lab, they do works in oysters and in coral reefs and in mangroves. We've done stuff in salt marshes and sponges, and we're actually starting to do some stuff in kelp. So I don't want you to think that all we do is play in seagrasses. But this is my shot to talk to some people, and with the opportunity I have, I'm going to focus the time I have on one habitat, which are seagrasses. So, the reason I'm going to do this is Did my battery die? I have the circle of death. Yep. YouTube sensation. Right here.
Right, because at this point he turns this into an interpretive dance.
Oh, man.
Just do the seagrass sing-along.
We could, and we might. All right. So, the title for my talk is "The Grass Withers and the Flowers Fade: A Global Crisis for Seagrass and One Group's Attempt to Try to Understand and Combat This." So before I start, I want you to put yourself into this picture for just a second. Okay? I actually want you to stand there. I want you to listen. You hear the birds, you hear the crickets, you see the rabbit kind of scurrying around, right? Going to his burrow, the deer on the edge, eating the grass. And imagine now how all of those interactions have changed by the loss of grass. Okay? And this story is going around all over the planet. People have become aware of this in the last decade or so. 2009, there was a paper where they looked at 215 sites, 11,000 observations at these different seagrass beds, and the red is telling you that they were declining. Okay? And when they started looking at this, we were losing about a football field of seagrass every 30 minutes. Total losses since 1880 was estimated to be about a third. So we lost about a third of the seagrasses. And what was particularly concerning was that the rate of decline was speeding up. We were losing more grass faster now than we had in the past. Well, what are seagrasses? Take a moment to just orient you. These are actual plants that are flowering. They have seeds. They have real roots. They started off life in the terrestrial world and then reinvaded the marine world. They also call them submerged aquatic vegetation. They're not seaweeds, and they're not algae. Now, like these land plants that have seeds, that got flowers, they are among the most productive of the marine environments. We'll talk about that in a second. And they provide a whole fleet of what we call ecosystem services, of which I'll mention a few of them to you. Because they're a plant, they require a lot of light, which causes them to have to grow in shallow water, which is adjacent to land, which is one of the reasons that they're in such peril. Now here's a seagrass that's been removed from the sediment. This is actually a terrestrial grass called Thalassia. But I want to point out a few things. They can grow clonally. They just add another shoot. And they're all attached along this thing called a rhizome. And here's one of those real roots that's getting sediments out of the soil. Now, being able to grow clonally like this allows them to grow to very, very large sizes and to live very, very long times. In fact, there was this paper that was looking at Posidonia, which is a seagrass in Spain, and they had measured clones, and they had measured that some of them had distances up to 15 kilometers. Same clone, 15 kilometers in length, and they estimated the ages of those clones to be somewhere between 30,000 and 100,000 years old, which put them to basically be dubbed the oldest living organism on the planet. Well, that caught the attention of this woman, Rachel Sussman, who was writing a book looking at the oldest living things on the planet. So she only looked at things that were older than 2,000 years. This is her taking a picture in the Posidonia beds. And she then produced this TED Talk, which I encourage you to look at. It's been viewed about 10 million times. But it put a lot of focus on seagrasses because of this long life clone. Not only do they grow clonally, but they also have sexual reproduction. When they invaded the world of the sea marine world about 100 million years ago, it was a very different place than it is now. You can see that there was much, much higher CO2 and a much, much higher temperature. Sea levels were much, much higher. Then over the next 60 million years, those dropped down. Well, because of their introduction and the time that they were introduced into the marine world, these black dots are showing you that there are seagrasses around every continent other than Antarctica. And certain species, can live in polar temperatures, and certain ones live in tropical temperatures, but we find them all around the planet, minus Antarctica. And if we look at this map for a second, it's showing you the diversity of seagrasses. And since there's a small crowd here, it's more like a class than a lecture. I'm going to ask some smart person right here to tell me, why do you think that there's so many species of seagrass in this particular part of the ocean? This is that long pause. What? Yeah, no.
Long-term stability.
Right? If we were looking at corals, we'd see the same exact thing, right? And so this is basically a very stable portion of the planet that's existed for a long period of time, allowing lots of diversification. You look at us over here on the East Coast, we only have 2 species along the East Coast. We have 6 to 7 species in the Gulf of Mexico. Here's just some examples of, they look very different. Posidonia has these wooden stems, look like little palm trees. These little ovate, Halophila, Rupia. So there's lots of different kind of growth forms for these seagrasses. Now, some of the ecosystem functions that they perform. If I were to ask you, before you came and sat on a chair here, which of these 3 habitats would you expect to be the most productive? Most of you would have said tropical rainforest, but some of you would have been thinking to yourself, wait a minute, I bet man could engineer plants to grow faster with fertilizer and genetics. But actually, the truth of the matter is that our eelgrass meadows along the coast of Long Island are more productive than either of those habitats are. And because these grasses stick up off the bottom, lots of things go and live in them and hide in them. Think of yourself driving down 27 at night, you see that deer on the side of the road and you scare it. Hopefully it doesn't run in front of you, but it runs into the woods. And the reason it runs into the woods is because it wants a sense of security. And so the same is true for the fish that are running into, and the crabs, the seagrass. These blades also have things that grow on them. We call them plants that grow on plants, or epiphytes. And they're a rich food resource for lots of things that grow up inside those seagrass beds. And therefore, grass beds have been known as serving a very important role as a nursery for most of the finfish and shellfish that we think of that we want to have, economically important ones. Now, these plants, they have roots and rhizomes in the soil, and like any plant, they photosynthesize, and so they basically pump oxygen in the sediment during the day, and at night they respire like any plant. They suck that oxygen back up again. So they're basically breathing oxygen in and out of the sediment every day. And that has a huge impact on the nutrient cycling that's going on underneath these grasses and the organisms that live in the sediments associated with these seagrasses. And they have been foraged by a lot of things, important foraging grounds. One thing I do want to mention to you, though, is this idea here, these Brant geese. Imagine this Brant geese landing in New Hampshire in a seagrass bed, and he eats the seeds and the leaves of this plant. He gets up and he flies to Long Island. Okay, now after a good meal in New Hampshire, and he lands in Long Island, his stomach's kind of full, and he voids out his stomach there in Long Island. And with it, all of these seeds that he had picked up in New Hampshire. Imagine millions of these birds flying up and down the East Coast, and what impact that had in kind of keeping the genetic stock mixed up. Well, we have less than a 18th of the Brant geese that we once had before. So that role is ended. Another really important service that they play, is that these roots and rhizomes, they help bind the sediment. And so they're really important in preventing erosion. And I have this picture here to think about fast-moving water, these strap-like leaves just laying down and capping that sediment, preventing it from being resuspended in heavy-moving water, fast-moving water. So keeping it from looking something like this. So they play a really important role in stability, and of course, they're foraged by some of the things. Now, this is kind of a... Does anybody know how these two animals are able to eat seagrass? Because I'll tell you right now, seagrass is full of lignin, which is hard to digest. What's your guess? How come these guys can do it? Well, they have multiple stomachs. Okay? Remember, these organisms also were in the terrestrial world and reinvaded the marine world. And so they basically can handle it because they have these multiple stomachs like a cow does on land. These animals right here, though, also reinvaded the marine world. They don't have multiple stomachs, but they have very acidic stomachs. And so they can break down that very undigestible cellulose in those grasses. Parrotfish can do this because they've got this crop that can help mechanically break down that. Anyway, because of all the services that I've talked about, seagrasses were valued 3 times higher than coral reefs. And most of you probably would not have thought that before you came into the room. Now, truth in advertising, as I was searching through all of the ecosystem services that seagrass play, I did find one where they ranked right at the bottom. So seagrasses ranks lowest in marine ecosystems in terms of inspiration for song lyrics. Now, right when you were to the point where you thought, I couldn't fall in love with seagrass more, there are 2 other things that I want to talk to you about. It may be shocking to some of you, but carbon dioxide is actually building up in our atmosphere. And when we look at the ice record, we see it increasing, and then since 1960, we've actually been measuring it in the atmosphere. And if we pay just attention to that record since the 1960s, can anybody tell me why that line goes like this?
Because there's more trees in the northern hemisphere.
That is the oscillation of the season. And what you're looking at is when the northern hemisphere plants are photosynthesizing, they draw that CO2 down, and in the winter it's respired. So that's what's causing it. It's plant production, productivity, sucking down that CO2. Now, there's a lot of people trying to figure out what they're going to do with all this carbon in the atmosphere. And some people have been looking towards the ocean as a way of storing some of this carbon. This article that was, actually was a workbook that was created in 2009 for the UN over in the EU was looking at the healthy role of the oceans in binding carbon. Interesting, in 1981, back when I was in 8th grade, go over, what grade were you in 1981? Sixth grade? There was this article that came out in Science that said marine macrophytes as a global carbon sink. Nobody cared about carbon back then, but they had realized the role that it was playing. So blue carbon is this idea of carbon sequestered and stored by the ocean, and the major players are seagrasses, tidal marshes, and mangroves. Now, pay attention. These three habitats only exist on 0.2% of the marine footprint. Okay? They only cover 0.2% of the ocean, and yet they're responsible for 50% of the carbon sequestration. And when we look at those three habitats compared to terrestrial habitats, here's tropical forest, boreal forest, temperate forest, and then those other three, you can see that there's no question. 40 times faster sequestration than forests, and these are long-term sinks. Now, think with me for a second, though. Why might that make sense? I mean, you're thinking of a tropical rainforest, right, or a temperate forest, lots of trees, bringing this carbon in, but why doesn't that carbon actually stay in those trees? I can think of a couple reasons. Fire, big time. Helps break that down. You know, if I had a time-lapse video of decomposition of a log here, you could see how quickly that biomass is broken down by other organisms. Now, someone tried to figure out how much carbon is actually bound up into the seagrasses, and a group basically came up with a number. They said 19.1 pentagrams of organic carbon. How much is a pentagram? It's hard to see. If we think of this 33-foot sphere as 1 ton of carbon. So I went to the web and I stole a video from Climate Visuals, and I hope it's going to work. This is for New York City in 1 day, 1 hour, 1 week. Citizen produces 15 tons of CO2 a year. If you're a vegetarian, you can take 0.8 tons away from that. I did not calculate this. I read this by somebody else. But if you make love 2 hours a week with the lights off, you can take 0.9%. 0.9. If you have a child, though, if you have a baby, you add 10,000. So, anyway, seagrasses have 19, almost 20 trillion of those bubbles locked up inside their below-ground biomass. Right? And with our losses, we see that we're basically 6 times that pile that you just saw in that movie is being lost every year because of seagrass loss. When we compare kind of your poster child for terrestrial, which would be your pristine Amazon forest, it's about 1 ton of carbon per hectare per year versus 17 times that for those Posidonia beds. This was so important that the IPCC added a supplement that focused specifically on wetlands, and they mentioned specifically the impacted role of seagrasses in carbon sequestration. And this has kind of spawned a number of initiatives, one being the Blue Carbon Initiative, which is looking at ways that we can basically use restoration of these habitats to drive carbon down. And in fact, this is really being led by the Australians. Their government is all in on this. Now, at the same time that CO2 is going up, the amount of CO2 in the water, seawater, is going up, and the pH then of that seawater is going down. So since the Industrial Revolution, we've seen an increase in CO2, an increase in temperature, and a decrease in pH units, and that's predicted to be even more so in the coming century. Now, Dr. Gobler was looking at the impact that this had on shellfish, and what you're seeing is basically the survival of shellfish as we increase the pH of these larvae are growing in. And that's got us thinking about seagrasses as oases. So this is, pay attention, this is big money's coming here. Oasis, ocean acidification sanctuaries and subsidies. That's clever. The point here is that the plants are photosynthesizing, driving down that CO2 and raising that temperature, making them a sanctuary from this ocean acidification. And the question is, does this fluid underneath that canopy, does it move out with the tide? And if so, how far does it extend? Does it add a subsidy outside of its boundaries? So, Allie has started looking at this, and this is one of Chris's graduate students, and he has all these fancy instruments. And I'm actually standing there with a wand that's pumping water up from the about 10 centimeters off the bottom, and it's going through all of these sensors, and he's watching the output on that as we walk across the seagrass. And so he basically says, "Are you in grass?" And I say, "Yes." "Are you in grass?" I say, "Yes." He goes, "Oh, you stepped out of grass." I go, "Yeah," because he can watch that impact of the drawdown of CO2 as I walk across the landscape. Now, why are we losing all this seagrass? That's the question. And there's a number of answers to this, but the common theme around the planet is algal biomass. Either it's macroalgae or in the water column. Well, what's driving this algal biomass increase? Well, it's not that hard. It's nutrient pollution. I like this cartoon. What do I need for a realistic marine environment? Well, runoff, coastal overdevelopment, unprocessed sewage. That's all you need. When we look at the world population, we see that it's just skyrocketed, predicted to be over 8 billion by 2025. And important for seagrasses is that our population is a coastal population. All the reds are places where there's 70 million or above people, and the oranges are 29 to 70 million within the coast, 100 meters, 100 kilometers of the coast there. And if we plot it this way, you can basically see that the dominant number of population really is within the 5 to 10 kilometer distance from the coast. So, all of these people are adding all of this nutrient. And this leads us to the Liebling law of the limited, which basically is stating that productivity is limited by one thing. And these boards around this bucket are those one things. And so in this example, nitrogen, is limiting. If we add nitrogen, we're going to increase the amount of water in our bucket. We look at experiments where they've done things like macroalgae, where you control versus nitrogen. You see that macroalgae is much more robust when we add the nitrogen, or the phosphorus, much more robust than when it's not there. So they are facilitating this growth. If we look at this impact of nutrient enrichment on seagrass, this is the sweet spot. Seagrass is happy, but as we increase, things start growing on the leaves, or macroalgae grows. And if we increase it too much, so much stuff is growing in the water column that there's no light reaching the bottom. And so seagrasses are kind of like that canary in the mine, because seagrasses require, this is percent surface light, they require 10 to 30% of the light reaching the surface. And when you compare it to the other marine plants like phytoplankton, algae, it's a magnitude of order more. So they're the first thing that seems to disappear with this over-enrichment of nutrients. Now this plot is pointing out 70 different estuaries around the globe. And they're plotting them as eelgrass loss as a function of nitrogen loading. And the things that stick out in my mind is that once we get above 175, we basically lose all of our seagrass. But when we look at just, take 50 for a second. You can see that we have loss down here, 5%, or we could have a loss of 100% at that same loading rate. And so I was always interested, why? Why is there such variation in loss as a function of this? But what about us? What about Long Island? We've got two species of grass. We've got rupia and we've got eelgrass. And we've got lots of critters that live here. Most of you are familiar with our bay scallops, which use seagrasses as a place to stay off the bottom. But lots of important finfish also locally use them. We've lost seagrasses in the Peconic. If you look at this figure from Cornell, all the blue is where there was grass in the 1930s. The red is where there's grass existing now. In the South Shore estuaries, in the 1930s, we had a wasting disease. We lost roughly 90% of the seagrass because of that wasting disease. And that was in the northern hemisphere. In 67 to 77, we lost another 23%. That was primarily from increasing nutrient enrichment. In the 80s, we lost another 40% because of brown tide. And the question is, what's happened since then as far as our losses of our seagrasses in the South Shore estuaries? And that's the question mark. So Chris, in his introduction, talked about us doing things together. In 2004, Chris and I got Sea Grant. And the question of the Sea Grant was, what would happen if we had a barrier breach through the island? That's kind of prophetic, given what happened several years ago. But what we did is we used inlets as our starting point. And the farther we got away from the inlet, we saw the chlorophyll level go up. But even more importantly than the chlorophyll level going up, it became more and more composed of smaller and smaller particles, things that are more difficult to feed on. At the same time, the seagrass productivity went down. So more stuff in the water column above them, shading them, reduced their production. I was thinking then about what role this might play and what has happened since then. Well, this is a picture that I probably stole from Chris, because he likes to talk about in the 1970s, 2 out of every 3 hard clams were consumed on the east coast of the United States came from Great South Bay. And this is what one of those boats would have looked like. Well, since those peak landings, hard clams really were decimated. Their populations became so low because they fertilized by sperm and egg in the water column. You can imagine if they're close together, their fertilization succession is good, but the farther and farther apart they get, the less successful they become. So their density became so low that they were really unable to recover from that since then. So in 1976, 700,000 bushels were taken out of the Great South Bay from these 3 towns. And if we do a number on the back of the calculator, if we estimate the filtration rate of clams and how big the volume of Great South Bay was, it basically took 3 days for the entire volume of Great South Bay, if we treat it like a bathtub, to go through the gut of a hard clam. We do it today, it's now 108 days. So there's real no biotic control of that algae. And in 1985, we had the first brown tide. So this was kind of what the South Shore estuaries looked like. Low phytoplankton biomass, lots of filtration, lots of light, lots of grass. And this is the current situation, another alternative stable state. So the question is, what do we do now? Right? Well, in 2007, the governor appointed a task force, Seagrass task force. And you'll see that, there's Dr. Goldberg right there looking so young. We brought in a number of seagrass experts from outside the state of New York to basically ask the question, how did we get here, and what can we do to push it back in that direction? Well, one of the questions that came up from that was, what about your diversity in your seagrasses? So let's just think about species now for a second, species diversity. Biodiversity as an insurance. So you can imagine we have two different kinds of species. This guy, he can live in cold years but not warm years. This guy can live in warm years but not cold years. So as we go along, if we have a community that's just made up of this guy, in a warm year, he thins out. Cold year does good. Vice versa for that. But if they're together, then it doesn't really change. And that's the idea of this insurance of biodiversity. But this all hinges on the response diversity, the variability of traits that allows these organisms to change to these changing environments. So let's think about seagrass. We think about eelgrass for a second. There was work that was done by a guy named Roosh over in the Baltic. And what the top panel is showing you is that there was, as they increased the number of clones, genotypes, the mix that had the highest genotype had more shoots than the others, the low. So higher diversity led to more shoots per square meter. And if you look at each of those genotypes individually, you'll see that some did really well and some didn't do so hot. So if we think of genetic diversity now here being important. Another study that was done looking at resistance and then recovery, looking at monocultures, meaning only one species or groups of species of seagrasses. So the problem with monocultures is they're not very resistant. But in recovery, they actually can recover more quickly. So there was a paper that came out from a bunch of guys over in New Jersey, which basically stated that eelgrass genetic diversity on Long Island was really bad and it was a bottleneck. They were suggesting, based on their samples, that we only had Labyrinthula-resistant shade-adapted clones left in Long Island. And that was of concern. So we went out, we looked at genetic diversity all around Long Island, and we found that to be absolutely false. In fact, if we took more than 8 steps away from where we took a sample, we never found another clone. If we took 1 step away, the likelihood of finding a clone was about 10%. So we had lots and lots of genetic diversity in our seagrass beds, which basically is telling you that our populations are driven by seeds, not by clonal development. No evidence of this bottleneck. So Goldberg also did a study back in the brown tide days. So this is brown tide, no hard clams. This is brown tide with hard clams. You can basically see those hard clams actually had an impact on that brown tide, which got us thinking about, well, okay, what's the role then of this biotic filtration? So Chris and I had a PhD student together, and he looked at this idea of the grazing down and the clearing of the water. So it basically is based on things like this. I love this video. So you're looking at some hard clams there, filtering time-lapse over a 25-minute period. But you can see, you get the point, right? That this clarity was improved by them clearing out the water and consuming the algae. So Chuck put out these large mesocosms, a little over a meter deep, and he would vary the number of hard clams in them. And when he added more hard clams, the chlorophyll got less and less in those buckets. And as the chlorophyll got less and less, the productivity on the seagrass in the bottom of those buckets increased. And you can make this nice leaf, as we basically reduced the chlorophyll, we saw this linear impact on the productivity of the grass. So hard clams significantly increased seagrass productivity. And in fact, he did it with hard clams and oysters and blue mussels, and every time he did it, he saw this impact, except for once. These are all of his experiments. There was one time where he saw no impact, and that was because the turnover time in his mesocosms was greater than this 4-day limit. Which again, got us thinking about, remember Great South Bay, every 3 days, going through the gut of a hard clam. whenever he had it for less than 4 days, he saw this significant increase in seagrass productivity. So John Carroll came along, and he was asking the question, okay, so let's say that these hard clams, they can't affect the light level, but they're also adding this feces and pseudo-feces in the sediment around them. So it's kind of like you're having a bad day, right? And I give you a piece of chocolate. It doesn't really change your day, but it makes you feel better about yourself. And that's kind of the idea here, that adding this nutrient, okay, allowed them to basically exist under this stress. So he did his experiments. He shaded, and he added a hard clam, and he added one of those fertilizer spikes like you could use it for a shrub in your yard. So what I'm showing you here is basically if we shade the plant, those plants that were shaded didn't do so well as those plants that weren't shaded. And then the following ones are the shaded and non-shaded if we added a clam to them. And the point that I really want you to take from this is that the increase of adding that nutrient was much more important for those plants that were under stress than it was for those plants that weren't under stress. Okay, what about the impact of adding this nutrient resource on reproductive output of the plants? So Lisa then did this experiment where she fertilized seagrass patches. And what she found was that when she fertilized them, they grew larger reproductive shoots, and they had almost a doubling in the number of seeds per shoot that happened. And why might that be? Well, those longer shoots were able to capture perhaps pollen from outside the patches than those that were primarily getting fertilized from within the patches. That's a speculation. But what we do know is that the seed input was doubled. So, shellfish. How do we deal with clean water? The vacuums of the sea. Clam power. One thing that we're doing currently is we're creating a bio-optical model. That's a fancy word for we're measuring the live stuff in the water, chlorophyll, and the dead stuff in the water, what we call total suspended solids, and then the color of the water. But the kind of when you put your tea bag in a cup and it turns the water brown, that dissolved organic matter. And by knowing those three things, we can then understand what's driving the light characteristics and limiting the seagrass growth. And that's what Caitlin's doing right now for the Peconics and for the South Shore estuaries. Now, the Nature Conservancy, put out a bunch of spawner sanctuaries with this idea of putting them together. We're going to increase their spawner success. They met with limited success, unfortunately, because of the high density of whelk that were causing problems for them, as well as some other issues. But SHRP, which stands for Shinnecock Bay Restoration Project, was basically born from this idea of we know what the problem is. We have too much nitrogen coming from our groundwater in Shinnecock. We understand that. But turning that tap down is a political $10 billion answer. So Chris and I were thinking, well, what can we do right now? And right now what we can do is we can increase the biotic filtration and drive that nitrogen from the water column down, out. And so the Shinnecock Bay Restoration Program was born, and since its inception, a bunch of hard clams, a bunch of oysters. We're about ready to put out the very first Long Island oyster reefs. There's been some in the Hudson, but we're about ready to put 2 out, hopefully this year, maybe next spring. Now, here's a picture from Gobler looking at the brown tide densities in Shinnecock Bay since restoration kind of began. You can kind of see this trend, 2016, bad year. But we saw this decline. We're encouraged and excited about that. So the idea is that if these hard clams are kind of weighing down, they're fighting against this nutrient input, driving this back in this direction. So what I'm arguing to you is that seagrasses, they've got this idea, what we call a fundamental niche. This is a place where they could live. But decreased light, toxicity, increased water temperatures, that's all making where they actually live less than where they could. And shellfish, my hard clam here, is basically filtering the water, increasing the nutrient pool, oxygenating the sediment, which is allowing them to exist in areas that they would not be able to exist in if the shellfish wasn't present. All right, let's talk about restoration shortly. restoration. We want to bring seagrass back. You can imagine how many man-hours it would take to get 10,000 shoots. Right? You got to dig them out of the ground, and then you got to keep them alive, and then you got to replant them somewhere else. And some people bring sod, some people tie them onto these turf trays, some people plant them on rocks or create these tortillas out of them. But the amount of effort that it takes to put down 10,000 seeds is a lot, and that's why most people don't actually do that many shoots. This is a picture of restoration of seagrass around the planet, and what you can see, here's the bar, is that less than 100, 100 to 1,000, 1,000 to 10,000. For us, in the North Atlantic, vast majority of our restoration has been 100 to 1,000 shoots. And people who've gone and looked at restoration have noticed that when you do that kind of number of shoots, your ability, basically the survival is low. Actually, this is higher than most survival on Long Island. Most survival on Long Island of our restoration is about 15%. But low, which increases as you get more and more shoots out there. And this actually, the idea is the growth rate of those that you put down is actually vastly enhanced when you get higher. Now, to restore an acre of seagrass is estimated to cost roughly $1 million for an acre because of this hard effort that's put into making them go there. But that's not true for seeds. You can collect 6 million seeds with 2 weeks worth of work and 5 people collecting reproductive shoots. This is a picture from Bob Orth down in Virginia. What you're looking at is his boat track. So he was at the back of the boat throwing seeds off as the boat floated around, leaving this track. So we use seeds because seeds are easy. We go out in the field and we collect these reproductive shoots. We tie them into bags. We attach a float to them. We throw them off on a cinder block and they float around. And they release those seeds from those reproductive shoots. And we come back the next year and you can see these little circles underneath those buoys. Actually, 2 days ago, although Howard Wiseman stood me up, 2 days ago, we did go out and we put in seeds from 5 different populations around Long Island and the Great South Bay. And the idea was that we collected these different seeds from these different populations, we put them in these little burlap packets, identified them, and we stuck them in the sediment so that we could then see, are different populations of seeds in Long Island predisposed to do better in poor water quality than others? We'll go back in the spring and find out if there was any difference. Now, I sense a sea change, a change in the air. You may have heard that a month or so ago, Chris and I were able to go out on the boat with the governor and to talk to him a little bit about the Shinnecock Bay Restoration Project. And recently, he put up $10 million for shellfish restoration on Long Island in different areas to hopefully drive this nitrogen out of the water column. Now, we have plenty of examples around the planet that when we deal with our nitrogen and we deal with our phosphorus, that the Secchi depths go up, and eventually we see this increase in the depth limit of Zostera. But you should point out, there was a lot of dropping of these things before there was actually any change in that Zostera heading up. So this is not a short-term 2-year project. This is a project that's gone, this one was roughly 35 years of restoration. I want to leave you with this thought, okay? That if we can actually change the system, the plant itself has a vast ability from its seeds to then claim space. And so Brad used an aerial balloon to get some images of a place in Shinnecock. So we stole some images from the web. So the green is seagrass, a plot in Shinnecock. 2001, we also got an image from 2006, another image from 2007, and then we started taking them with the balloon in 2009. And so you'll see here's in August. You see that in March? August, March. What you're looking at is a great seed set that happened that year. And then in June, As it coalesced, August, dies back in December, May, August, December, April, July, December, May. So you've seen the sequence, but really, when we started off, less than 3% of the bottom had seagrass on it. By the end of it, over 43% of the bottom had seagrass, right? But that's from 2001 to 2014. Actually, if we just looked from 2009, we went from less than 8% to over 43% in 6 years, right? So I'm arguing that putting shoots in the bottom is not the strategy we need to have. What we need to have is a strategy that's going to change the system, because the plant itself has an ability to come. And we've seen that here locally in Long Island. Go Seagrass.
Questions for Dr. Peterson?
Yes.
When you said that seagrass, tidal marshes, and mangroves take up 0.2% of the coverage,
Yes.
50% of
Yes.
sequestration,
Yes.
is that sequestration into the ocean or just?
Into the sediment.
Okay.
So the idea is they can lock it up in the sediment. Usually it's anoxic sediment, which means there's no microbial breaking it down. And so it can be safe for millennia, as long as it's not opened up and exposed. Yeah.
And that last slide with that incredible expanse of seagrass over 6 years, what is the change in the system that allowed that?
So where you were looking at, what we're looking at was just a natural seed set that then grew and coalesced in Shinnecock. Water quality was great there, right? I'm hoping for that same thing to occur over on the other side of the curation in time as water goes up. So there was no improvement in that. That was just the natural progression. Yeah.
So you argue that it's a systemic change that is going to drive
Yes.
more?
Yeah. Because let's talk about, so everybody wants to do something. They see a problem, they want to do something, right? And so a lot of people say, we used to have grass here. And so we'll get grass, we'll put it back where it was. The problem is where it was is not the same as what it had been before. And so a lot of times, people put a lot of effort and money into putting this grass back in this place, and it dies. And how do we feel? Like, right? And so I think we need to be careful, because there's only so much money. There's only so much time we have. We need to make sure that we're investing in the best possible way to get success. Now, let me be very clear. I am not telling you that seeds will work everywhere. I'm not telling you that, okay? But what I'm telling you is that we have seen success in seeds in Long Island, in the South Shore estuaries, and if you can use seeds, think of this, 15% survival. I put 10,000 shoots down, 15%. How many do I have left? 1,500, right? I put 1,000,000 seeds down. I have 50% survival. I've got 150,000, right? One took 3 men a month. One took, 5 people in 2 weeks. So that's why I argue, if you can do it.
Okay.
So the plot you had, when you were showing the progression through time, that was like after some seeds were placed?
Natural seed set. Yep.
So you're just looking at plants that already existed and following them?
Yes, that's correct. I did nothing to those. We just observed it, right? But there is something that you should pay attention to that, right? There seems to be some kind of threshold density that when you get to, it actually changes the system, right? And so it's important that we continue on. I think you saw that a little bit in that restoration from the Danish, right? They continued on and they saw things go down, nothing happened, and all of a sudden, boom, that limit went up. And so my argument is that, continue to push. Our 2-year or 3-year idea of restoration is not, it didn't take 2 or 3 years to get us to where we are. It's not going to take us 2 or 3 years to get us back where we were. Yes, ma'am.
The experiment in Reproductive Bay with DNC,
Yeah.
red seeds down, is that all the same species or just from different areas of the island?
We talk about the seeds here. So these were different seed populations around Long Island. So we know of a couple places that are really bad water. So little time. So one idea of taking adult shoots, right, is we take adult shoots from here, we pull them out of the ground, and we put them into this other place, bad place, boom, put them in the ground. And the problem is that when that seed germinated, it started turning on genes and started growing in a particular habitat type, adding chlorophyll to its leaves, adding air pockets below ground in this sediment, in this place that it's at. Now I take it out of there and I put it over here, and it doesn't do so great because it's a totally different place. And so what we wanted to see was if we take seeds, all seeds from different places, put them all in the same condition, and see are some of them basically predisposed to do better than others in this bad water quality. Because if I take seeds from bad water, or shoots from bad water quality, and put them into bad water quality, they're going to do fine, because they grew up in bad water quality. If they shoots from good water quality, put them in bad quality, they're going to crap out.
But even though we only have one species of
Only one species, they're just populations, a different eelgrass
we do
population.
have a good genetic diversity.
Yep. Yep. You know, like when we first started talking about this, the guys in the Baltic thought we were crazy because they only have 5 reproductive shoots per square meter. We have over 40. So it's a very different system than on the other side of the Atlantic. Yeah.
What's the most common epiphyte or macroalgae on eelgrass in our waters?
Well, it changes with the season, but
Or, yeah.
do you know slip gut? You know what I'm talking about? Common name?
No, but I'll look it up.
Well, no, it's a common name. You're not probably not going to go find it. Can you guys give me a name for that? Yeah. Ectopurpose. So we see a transition as the season goes on, and so we can have some kind of particular parts, but then as it gets colder in the fall, we see the slip gut seems to get all over, and in the spring as well. But as the water warms up, that seems to go away. Something else. By the way, you guys are free to watch the game.
Naturally, I want to ask about the brants.
Yeah.
What happened to them? Why did their population?
We shot them.
Okay.
We ate them for food. But it really made me think a lot about avian bird consumption of this plant material because just imagine it going up, and they consume a lot. Swans will eat grass as well, but there's also there's another that I don't know that also would eat it as well. But brant geese are big consumers of it, and so they were the big major players. In fact, there's like in New Hampshire, there was a place where you can actually see where there was I guess, the way the pattern of the migration came, there was a lot more that came in one year than the other, and you could see kind of they defoliated the grass bed. But that's not common. Yes, sir.
Why not make, instead of eelgrass, make a monster grass? Make a genetically modified.
Well, listen, it's not that bizarre. People are doing that for coral right now. People are basically trying to genetically choose genes that can basically survive in high temperature and use those for restoration. We just recently got the entire genome of eelgrass done in the last few years. So
Oh, wow. That's a big step.
it is a big step, but I think we're far behind a lot of the other terrestrial folks in being able to do things like that. Yeah.
When you said that the population was driven by seeds, you're analyzing an area that when you took one step,
Yep.
you didn't see a clone.
Yep.
Was that from restoration projects, or was that
Nope.
just seeing like how they naturally produce?
Naturally.
So
That was
does
all.
mean they're under a high stress environment?
That's a
then?
really good question. That's a really good question, right? Because here's the deal. If you thought life was good, you would expect that, I would expect that clonal progression of the grass would occur when things are good, because theory would suggest that. And that they would only really have, they always have some sexual going on, but they really ramp that up only if they have to get out of the area, right? So, is that basically a precursor to our, are they overly stressed and they're putting out seeds because it's really bad?
Yeah, I was wondering like what those, how would you approach trying
Another
to
thing to keep in mind, and this one, I think we saw this with the breach. Seeds are only viable for 2 years. So you rain them out, and you got 2 bad years, all the seeds are gone. And so we had the breach, we had great cool water, clear water come in, right? And I got out there, I was ready, we're going to see it happen. And the first year, nothing really happened, right? The second year, a couple patches. The third year, they popped up in a lot of places. I mean, the size of this room popped up in places. But I think it took time for those seeds to kind of move from the population to places and sprout. Yes, sir.
So back in the 70s, I was spent a lot of time in Patchogue, the Great South Bay, and that eelgrass was washing up in mountains every
Yeah.
single day. I remember them removing it. But something else I remember then too, along with the clamming, was all the clam raking. So is the actual physical raking taking away the eelgrass also, or is that negligible compared to the other impacts from the waves?
So first, most guys who know what they're doing don't want to be in a grass bed because they basically, it fouls up their rake and it's a hard thing to do. People recreational that do it will get the above ground biomass, the leaves off, but the leaves can grow back. As long as we're not tearing into the bottom, we're okay. But these rhizomes aren't very deep for eelgrass, so it is easy to get them up and going. Most of the baymen guys, they want to get on the edge of the grass bed. They want to get into the grass bed. But you can go to places in Great South Bay and you can fly your little drone and you'll see lots of places where people have driven boats in shallow water and made tracks where they cut through the bottom and knocked it up. I imagine that the same thing could happen by someone in the bridge if they were to spot like that. Yeah.
Does the rate of compaction of the sediment have a major role on the rate of growth in terms of changing it as the sediment compacts and there's less suspended material?
Yeah. So I don't know if I can answer your question. What I can tell you is that, like when you walk into a forest on a windy day, you don't feel the wind. Same as the water moves through these seagrasses, it slows down and particles are falling out. And so the rate of particles inside grass is along the edge of the grasses is higher than outside of that. And that builds up organic matter, which builds up sulfide and issues like that. So the compaction, I don't know. What I can tell you is that when they had the oil spill in the Gulf, one of the problems they had was that the oil removed the surface tension between the particles of sediment. And so it was very easy to pull these plants out of the sediment when the oil was there, which is the reverse of what you're asking.
Right, well, please join me in thanking Dr. Peterson.
Thanks for coming.